Hello and welcome. So this is a lecture on water movement in plants. Okay, so we talked last time about the fact that water moves from high water potential to lower water potentials, less negative to more negative. Today we'll talk a little bit about the soil, the properties of soil that influence the ability of plants to extract water from the soil, and then later also the effects that the humidity or the air surrounding the plant has on water flow through the plant. Okay, so effects of soil properties on water movement. So let's first look at solutes. So we know that solutes inside a solution will create a more negative solute potential. So in soils, the solute or the osmotic potential is often near zero, but it can be lower than that. It can be lower in salty soils. So if a soil has a negative solute potential, more negative than the plant, then water actually moves out of the plant into the soil. That's, of course, a big problem for agriculture, for plants. Next thing that's important in the movement of water through the soil into the plant is the soil particle size. So we have sand, for example, with larger particles, clay with smaller particles. That affects the surface area of particles, as you'll see in this table that I'll show you in a second. And then the pressure potential of the soil. Just like a cell that we looked at, where we said there's the pressure potential of the cell that has something to do with the turgor and the amount of pressure on the outside of the cell. There's pressure potential in the soil, and that is usually zero when the soil is wet. That means the water isn't really moving anywhere. But when it's drying out, when the soil is drying out, it can become very negative. It can become so negative that the soil acts like a sponge, and it, as soon as you add a little bit of water to that soil, it will move away the water into the soil further, but not into the plant. And a important way to describe how water is held by soil is the water holding capacity. So that is the ability of this soil to hold water without it moving right away, right through the soil. So sand, for example, holds very little water. You pour water into a flower pot full of sand and it runs straight through, while clay holds a lot of water. And potting soil, organic material, for example, is somewhere in between. So it holds a little bit more water than sand, but less than clay. So there are several ways to describe soil properties, and one of them is the field capacity, and that is the amount of water, the water content of a saturated soil. So think about a certain amount of soil that you have that's dry, and the amount of water that you need until it is completely saturated and starts to drain out of the soil. Then you have reached the field capacity of that particular soil. And again, for sand, that's very low. For clay or for organic material, potting soil and so forth, it's much higher. And the soil hydraulic conductivity is something else we'll also discuss in a second here. That is a unit that depends on the ease of which water can flow through soil. So let's take a look at that. So the field capacity of soil, I said that there are some differences here in the particle sizes. So you see sand, for example, coarse and fine sand, you see the particle size here in micrometers. And if you have large particle sizes, then the surface area per gram of this material is lower than if you have smaller particle sizes with the same amount of gram, giving you a much greater surface area. And that's important with respect to nutrients, ions, and so forth being held by the soil. We'll get to that when we talk about nutrients in nutrient uptake into the plant. So the soil hydraulic conductivity, you can see here in this figure, here's the root and here's water. Air pockets inside the soil solution will slow down water movement. And sand particles or clay particles have a different affinity for water. And so they can hold water more closely to this particle, or less closely, or less strongly, I should say. And so therefore that will affect how fast or how well water can move through the soil as well. And so you can see that will affect whether or not a plant can pick up water from the soil as well. All right, so now let's get here to this cross-section of a plant and see how water moves into the plant. So this part here, this entire part here, that's called the stele, S-T-E-L-E, the stele. That's the inside of the plant root. And so you see here the xylem, the blue parts, that's the phloem. So we saw that in the cross sections in the lab. And then this part here is the Casparian strip. So we'll talk more about the Casparian strip. But the red parts here, you see, if you look at this in this blow up, you see that it covers the outside. It's like a circle, like a band around the cell, but not the front and the back. So what that is this Casparian strip is a suberin material. So it's a waxy material. It's called suberin. This here, suberin, and that is a waxy material, so that sort of impregnates the cell against water. Now, why is that important? Why is that advantageous for the plant? Well, that is because if water is supposed to be moved into the xylem and then up into the plant, it needs to get from the outside, from the cell walls, where it can stay basically without ever entering into a cell. It can sort of stay in the cell wall material that's, like a wick. It holds water, but it doesn't require, there's no need for the water to be going through the membrane, which requires more energy than just to be going through a place where it's basically pulled. So that doesn't help the plant if the water never enters into the cell. And so at some point it has to go through the membrane into the cell. And you see that it does that here. And then it's in the symplasm of the plant. So the inside of the plant cells. There it then can get entered into the xylem, and from there can get moved into through the stem, into the leaves, and so forth. So that is the symplastic pathway. That is the symplastic pathway when it gets into the cell, and it does that at this point here when it enters into the stele through this part of the cell, and the Casparian strip, this waxy layer, prevents that water can just remain in the cell wall. So here it cannot go any further because it's prevented from further movement by this waxy strip, by this Casparian strip. That means it has to go around and has to go through the membrane and enter into the symplasm. Now, you see here the symplastic and transmembrane pathways. Here you see that it's also possible for water to immediately move into the symplasm. So if it goes through the cell wall here and into the through the cell membrane, then into the cell, it can go through these through the symplastic pathway from cell to cell into the stele. So we have the apoplastic pathway and the symplastic pathway. Apoplast is the intercellular space, the cell walls and the xylem vessels. And then the symplast is the total protoplasmic volume of the plant except the guard cells. So that's the definition of the symplast and the apoplast. And now I said that in the symplast, to get into the symplast, it has to get into the xylem. That is correct. And this part here from in here is symplastic, but the xylem vessels themselves are excluded by definition from the apoplast because they are hollow, they're dead, they're not really living cells. They're just tubes, water tubes. So the water does have to go through the symplast here, and then from there on, yes, it goes back into the apoplast here if it zips up into the leaves, but then from there it enters back into the symplast. So basically symplast inside the cells, apoplast outside the cells. And then the Casparian strip, here's the suberin. Those are that is the part that forces the water to enter the symplasm. All right, so water will move from the soil into a root as long as the root has a lower water potential than the soil, right? And that is thermodynamics. We talked about how that happens when there is a lower water potential versus a higher water potential. So the plant just needs to keep a lower water potential inside the root for it to be able to pick up water from the soil. All right, so let's look at a phenomenon. We'll come back to the root in a second. That you sometimes see on plants, and what you see here. This is not dewdrops that you can also sometimes see on a plant. These are guttation droplets, and what that is water that is being extruded out of the leaves. Not all plants have that, not all leaves have this, but some have these specialized cells that are called hydrathodes. And so excess water can be excreted from the leaves out into the ocean, into the open, where it then can evaporate. So we'll come back to that, how water can be picked up, but when there's an excess of water, it doesn't get excreted through the roots, it gets excreted through the leaves. And this can help causing something that's called root pressure. So let's look at how that works. If you have a low solute potential in the roots, for example, in the root xylem, then you can have water streaming into the inside of the symplasm of the root. So water is moving into the root because it's saltier inside, essentially. And that creates high water concentration, right? So there's more water now inside the root. And that creates something that's called root pressure. So when you have root pressure, you actually increase the amount of pressure inside the cells because there's just so much water, right? So now you have turgor because water keeps streaming in because it's saltier inside. And so unless the pressure from the cell walls becomes greater than the suction, for the water to go into the cells, you will build up pressure inside these cells. And since there is an outlet, if you will, for the water as the water travels up the plant, the stem, and into the leaves, that root pressure helps build that water stream up into the plant. But when you don't have transpiration, for example, at night, then this root pressure can also lead to the fact that there's excess water, and that water then gets pressed out through these hydrathodes. So increased even more by the lack of transpiration, for example, due to high relative humidity, which you often have early in the mornings, lack of leaves. You can sometimes see that as well with a stump. If you look at a tree stump, you can see water that still comes out of the stumps. That's of course not guttation; those are not hydrathodes there. This is just an example of root pressure. And if you have closed stomata, then water cannot transpire through the leaves, and in that case, you can have guttation as well. But all that comes from the root pressure, and of course, all that comes from the fact that there is a lower solute potential in the root, and therefore more water moving into the roots. All right, so now let's think about what might influence transpiration. So transpiration is usually influenced by environmental conditions, temperature, wind, light intensity too, because light intensity has something to do with the stomata. So those are the little pores in the leaf that open or close. We get back to that when we talk about photosynthesis and why and how the plant regulates its stomatal opening. But those are environmental conditions that can change and that can affect transpiration. And then we have the leaf-air interface. That's called the leaf boundary layer. We'll take a look at that a little more closely here. That can also affect how much water can transpire out of the leaves. All right, so here's a leaf. And this is the boundary layer, the air boundary layer. But let's first look at the rest of the leaf. So we've looked at cross-section of leaves before. Here's our leaf. Here's a cross-section. That is the bit that we're looking at here. Here's the palisade parenchyma. Here are the mesophyll cells, spongy mesophyll. Here's a cross-section through xylem and phloem. So this is the vascular tissue here, vascular bundle, lower epidermis. Here are stomata, so these are the openings in the leaf through which the water can evaporate out, transpire out, and carbon dioxide can come into the cell. And this part here is the leaf boundary. And so when you have a boundary layer, that creates resistance for water to transpire. So think about it this way. If you have an animal, let's say you think about an animal and you think about a cold day, the way or the reason for why an animal can withstand cold, many animals at least, because they have a fur, they have a big, thick fur. And so in that thick fur, temperature, the heat is trapped, and wind cannot penetrate into the down to the skin and cannot take the heat away as easily. And the same thing is true here. So here we have a boundary layer, and that boundary layer we'll talk about a little bit in more detail as well, what makes up that boundary layer. But if there's a boundary layer where wind cannot blow away humidity as easily, then you have a greater concentration of water vapor just around the leaf, and doesn't get blown away if the boundary layer can prevent that. If it's completely naked, if there is just, if there's a flat surface and there's a bit of a wind, then the wind will immediately blow away the moisture, and then it makes it for a drier area right out here, and that means that more water is transpiring out. So more wind, less boundary layer, more water transpiration, and so therefore more loss of water. All right, so let's sum this up here a little bit. The boundary layer resistance is determined by the wind speed. Faster wind means smaller boundary layer. The leaf size and shape. Some of the leaves that you might have seen, they start to roll up in the winter when it gets cold. Rhododendrons, for example, will do that. When it gets really cold, you look out, you see that they're rolled up. That helps them with increasing the boundary layer. Grass leaves are smaller. They're sometimes a little. They're not just a blade. They look a little bit V-shaped, and sometimes they're rolled a little bit. The presence of trichomes, so those are leaf hairs. Some leaves are very hairy. Some are very unhairy. Corn leaves, for example, don't have very many leaves and not very many hairs on them. But if you go up into the mountains, you see that where it's colder, oftentimes plants are sort of fuzzy. They have more hairs, and that helps them with the boundary layer and to increase the boundary layer resistance. And then there can also be things like sunken stomata. I'll show you a cross-section of some sunken stomata in a second. So in a sunken stomata there, you have you create a little cavity where the water gets trapped after it's been transpired, and so therefore it increases the water concentration out there, which means that water is less likely to transpire out of the stoma. Okay, but I keep talking about the stomata, and I know that you've probably heard about stomata before. I know we talk about that in Bio111. We look at that under the microscope, but let's just review what stomata are. Stomatal complexes consist of guard cells. So those are the cells that guard the stoma. Stoma just means mouth in Greek. So it's the opening usually on the underside of the leaf. Some species also have them on the upper side of the leaf. There are in some species so-called subsidiary cells. I'll show you a picture in a second. And then of course there's the stomatal pore in the middle. It's the opening between the guard cells. The guard cells guard how wide open that pore is. The further it's open, the greater the likelihood that water is lost. All right, so here are two types of stomata. This is the one with just the regular, with the guard cells and the opening here. And here's a type in a different species, for example. These are the guard cells, and these are the subsidiary cells. But in both cases, these look a little bit different from these. the guard cells here. But in both cases, what's important is that we have these radially arranged cellulose microfibrils. So that is basically, if you think about these pores like a balloon, like a party balloon that you blow up, or you can blow it up and it will just become a round shape. But if you put some restraints on the outside of this cell, or in this case, it's in the cell walls, these are the cellulose microfibrils, radially arranged. So coming back to the balloon, if you were to tape some duct tape on certain parts of the balloon and you blow it up, well, then that part is constrained. It cannot expand, right? Because the duct tape sits on there. So then you blow up this balloon, and it goes, it gets much bigger and rounder at the part where there isn't any constraint. And so that is what allows, in the case of the party balloon, the shape not to become just round. And in the case of these guard cells, that when they get filled with water, that they will sort of not just bulge out, but actually be prevented from bulging right here and right here. And that allows the opening of the stoma. So because they're constrained here and because they're constrained here, they cannot, they actually open up. And when they are flat, when there's no water around in the plant, when the plant needs to close the stomata, then they just sort of collapse. And then when they collapse, they will close this pore here. So that's how stomata work. Here's some examples. So this is an electron micrographs. This is a transmission electron micrographs, and these are scanning electron micrographs. This is another transmission electron micrograph. So here you see the cell. Here you see the guard cells here and here. Here you see the stomata, the stoma in between. Here you see a stoma between two guard cells. Right here is this one guard cell, and the other guard cell is down here. And here you have a transverse section. So the stoma is right here. That's where the air goes through, or the water transpires out. And you see that these cells are thickened here, they're thickened here, thickened here and there, but not so much here. And so that changes their ability and their way to expand when they're full of water. All right, so those are stomata. So important to remember is that you have high water vapor here. So this is high water vapor. And out here, it's low water vapor, right? So that is, that's the difference between the inside and the outside. And so since there's lower concentration of water vapor, the water potential is more negative on the outside. And so therefore, just like in the plant or in the soil, the water moves towards the lower potential. And so it will move out of the plant. On the other hand, here on this side, when we talk about, of course, this could be the same stoma, right? As they just drew it in two different places so that they wouldn't be conflicting with each other. Here we have carbon dioxide. And of course, here we have high concentration of carbon dioxide on the outside and the low concentration of carbon dioxide on the inside. And so the concentration gradient there is the opposite. Okay, so that part needs to be explained a little bit more. The water potential between the outside of the leaf and the inside of the leaf. And I'm going to do that in a second here. But I said I was going to show you some cross sections of stomata. So here's a sunken stoma. You see that this is the outside of the leaf. Here's the outside of the leaf. This is a cross section. Here's the palisade parenchyma, spongy parenchyma. Here's a vascular bundle. And so the outside of the leaf really is here, right? This is this part here. That's the outside of the leaf. Sort of like a bay, if you were to look at it, if you think about it as a map. And so water would transpire out here, out of the stomata. And I can't really see the stomata directly. Maybe there's one here. I don't know, maybe there's one here or so. But there's somewhere in here between these epidermal cells. And so then there's high water vapor concentration inside the leaf. And there's lower water vapor concentration in the sunken stoma, but it's still higher than here outside. And so that helps prevent the transpiration. So this would be an adaptation to occasionally dry climates. And here you see sunken stomata. So here you have a very thick outer membrane, right? So you see these thickenings here. You see that there's some collenchyma here, and then here are the stomata. So somewhere in here, maybe that's a stoma. Maybe there's a stoma here. So here, I guess those are the two guard cells there and there, here and there. But they're sunken in that they're not here on the outside. They're not there. They're further sunken in. That adds a little bit of boundary resistance layer right here. All right, so that's again adaptation to dry climate of some. So this is a conifer of some sort. All right, so we talked about stomata, we talked about water movement, we talked about water movement from the soil into the plant, into the leaf, from the leaf out into the air. And I said that the stomata have certain functions that deal with water vapor diffusion out and carbon dioxide into the plant. And as the carbon dioxide goes in, the oxygen from the photosynthesis can of course also leave the cell. So we talk about that in more detail when we talk about photosynthesis. Okay, so I said we wanted to talk a little bit more about the water movement from the cell, from the inside of the leaves into the atmosphere. So for that, what is important is how dry the air is. And the value that goes with that is the relative humidity. So it's something you hear on the radio when they talk about the weather. High relative humidity, of course, means that it's moist air, right? So muggy day has very high relative humidity. And what that is, the relative humidity, that is the current amount of water in the air divided by the maximum amount of water that could be in the air of the same temperature. For that, it's important to know that warm temperature can hold, air of warm temperature can hold more water than cold air. So when you have a certain amount of water that is held in warm air and that warm air cools, then that water is released. And that usually happens in the form of dew. So it condenses on surfaces. But regardless of what it is, the amount of water that's in the air at a given time can be more or less. But air can only hold so much water, and that is where this maximum amount of water that could be held at, let's say, 10 degrees Celsius versus the amount of water that you currently have in there. So let's say you have, let's just say 5 milliliters are in the air currently. 10 milliliters could be held in the air per a given volume at a given temperature. So then the relative humidity would be 50%. All right, so look at this table. This is really quite interesting, I think. So the relative humidity, when the relative humidity is 100%, that means it's really, really muggy outside, then the water potential of the air, now we're talking about the water potential of the air. Previously, we talked about water potential of a plant or even of the soil, but now we're talking about the water potential of the air. So the water potential of the air at 20 degrees Celsius at 100% relative humidity is 0. That means at that point, with really high relative humidity, plants do not transpire water anymore. That can be a problem because when plants cannot transpire water, just like when humans cannot transpire at a high temperature, humans or animals, well, let's talk about humans here, they will cool their body temperature, they will cool their outside through evaporative cooling by putting out sweat. And therefore, then when that evaporates, that takes up some energy, and so that cools the surface. And that's in fact also what happens in plants. So they do need the transpiration for the same reasons as humans do, for many other reasons as well, but it is one of the reasons that they do use it. And so when there is high relative humidity, there is no evaporative cooling that goes on because there's no suction, if you will, of the air, off the air from the water. So there's no water that gets transpired. But if you look at just a little bit of a drop here to 95% relative humidity, the negative value of the water potential of the air already drops to -7. That is much, much lower than quite a concentration of salt would be required to get that low in a cell. So -7 that's a low water potential. And then at really dry areas, in really dry areas where the air is really dry, the air is so dry that it really sucks the water out of a plant. It also sucks it out of a human, right? So that's why people always say if you go hiking and it's in a dry area, you need to drink a lot, because the air is so dry, it has such a low water potential that it really takes the water out of the organism. So the plant will have to come up with strategies to prevent water loss if it lives in areas that have very low relative humidity. Likewise, plants that live in 100% relative humidity will have to come up with adaptations so that the heat, if that comes at the same time, doesn't prevent them from doing their metabolic reactions. Okay, so let's talk a little bit about physical challenges to xylem water transport. So water that moves through the xylem from the roots to the leaves, and the problems that can arise there. So there is tension in the xylem, and what I mean with that is that there's a tension between water molecules. So you know that water molecules stick to each other because of hydrogen bonds in a certain way, but when they don't stick to each other, we saw that in the soil when I said that if there are air pockets, then the hydraulic conductivity goes down. Water doesn't move as far as well anymore. Then you get something that's called air seeding. So that can cause air bubbles, and these air bubbles can cause a loss of tension in the xylem. And with a loss of tension, and connectivity between water molecules, the water doesn't move up anymore. And if the water doesn't move up anymore, then the water doesn't get into the plant. And so the plant can wilt, even if it's standing in water, because it cannot pull up the water through the xylem, through into the leaves, and then out into the atmosphere, where the dryness of the air and the low water potential there causes the water movement through the plant. So air seeding, air bubbles can become a problem. Wounds in the xylem, leaks in the xylem, that can cause problems. Freezing temperatures can also lead to what's called degassing of water. So just like air can hold a certain amount of water at high temperatures versus lower temperatures, lower temperatures of water with lower temperatures can also hold less air, less gases that are dissolved. And so when you have freezing and you have degassing of water, then you get little air bubbles, small air bubbles coming out of the water. And they can introduce these bubbles in the xylem, and they can essentially cause embolism. So they can create a lack of tension in the xylem, and so then there's no movement from the water anymore, through the plant. So what do plants do to prevent that? Well, they can produce new xylem cells every year. So they just, when their cells have embolisms, then they just produce new ones. They can produce separate strands so that they're not dependent on one strand of xylem to move the water from the roots to the leaves. And they can create these, they grow these little vessel plates. I'll show you a picture of that in a second. They interrupt the vessels, so they basically put a ceiling from one story to the next story in there, so that the air bubbles they get trapped there. And so maybe that means the end of that one part of the vascular tissue of that xylem strand, but then at least the air bubble is trapped. And so that way they can prevent further movement of the air bubbles. They can also increase xylem pressure at night, and when they add the pressure, then that can redissolve the air into the water. So the degassing can be reversed by increasing pressure, and the pressure, of course, comes from the root pressure. We talked about that earlier. The root pressure comes from the fact that there is higher concentrations of solutes in the roots that lowers the solute potential, right? high concentration of solutes, lower soil potential. That means water moves in. And so there's so much water in the roots that causes pressure, and that pressure then can help with the redissolving of air and eliminating of bubbles. All right, so let me just show you here these cross sections. These are actually longitudinal sections of xylem tissue. So you see these things here. These are perforation plates. They have holes in them so that water can move through. The water moves from the bottom here up through to the top. So this is a perforation plate here. This is a xylem strand. These are two separate original cells. These are dead cells, right? There's nothing in there. They're hollow. But water can move through there. But air bubbles, when they get bigger than this perforation, then they get trapped here. And the same thing is true with these pits. So water can move through these pits, basically little holes, but maybe larger air bubbles cannot. So water flow through the xylem, as sort of a fun fact on the side, allows water to travel 10 to the 10 times faster than flow from cell to cell through membranes. So having vascular bundles allows plants to transport water so much more efficiently than without it. So that's an innovation that was done during evolution of plants with vessels, with vascular systems. Okay, and last slide here. So I want to show you a cell that has cavitated, where there's an air bubble in there. Here we have perforation plates, and now here we have multiple strands of xylem, right? So the water can still move here, moves through here, and sort of bypasses the end of the obstruction. So here's a pair of cells where you have a gas-filled intermediate tracheate. These are tracheates, are small vascular xylem elements. Here, tracheates, and vessel elements are the large ones. So these are vessel elements. It's just a different name for the different types of cells within the tissue. And then you see here, here's a perforation plate as well. So that's just basically a lid with holes that traps air bubbles but allows water to go through. And that's the end. That's the end of it. See you in class.